Are you going?
All right, so we get started, and then I'm only available Tuesday, and that's why I switched to the
I'll
lecture.
change all of them. I'm
Yeah,
sorry.
whenever no, I think I'm after done this have 2 more lectures, and then I will teach the new reality. Whenever Tuesday is my spot. Okay. So today we talk about molecular signaling using neurons. So basically, what we cover is the types of signaling and signaling molecules, receptor types. Spend some time with the G protein and the secondary messages, and the kinase and the phosphates and the nuclear signals. So that's the the topic we're going to cover. So what type of the cell communication is? This type is contact dependent. So what's the contact dependent? Is the signal molecular remains bound to the cell that produce it. Therefore, only influence the cell that directly contact it. And this is a very local type of signal. Is very important in developing multicellular organism and in the immune system. So this is the signaling cell, and this is the target, and this is the direct contact. The second type is what we call the paracrine signal. So what does this mean? And this is a target cell, and this is the molecular cell. Signal cell produce a signal molecular that is secreted. You see, this is a signal molecular, all right? So secreted, but only diffuse in very short distance. So this is, and then diffuse very short distance, the signal molecule acts as a local mediator that affects the cell only in the immediate environment of signal cells. So this is a single cell again. This is cell is secrete some signal. We have the local mediator. And because of the paracrine signal molecule act locally, their diffusion is limited. And the fact that limited diffusion are how rapid uptake by the neighboring target cell, how they disrupted by extracellular enzymes, how or by the immunization of the extracellular the matrix. So this is first type and this second type of the signaling there. So this is the target cell. This is mainly local. So we spend a lot of time in synapse, am I right? Synapse we talk a lot. I talked about, Dr. Newman talked about. So we have the neuron, we have the axon, we have the synapse. So this is everyone should be know. This is very basic, calcium flows in, release, and the target cell. Am I right? So this we spend a lot of time talking about this. So what is the synaptic specialized cell called the neuron? Makes the long process axon contact the cell far away. Neuron stimulated. It depends on the electrical impulse, action potential when the sodium open, when potassium, open, target cell. Reach the end of the axon, promote release of the chemicals called the neurotransmitter, the most famous one is acetylcholine, right? And then we have the GABA, we have the NMDA, we have etc. We talked about this 2-3 weeks ago. So this diffuses a very short distance to the target cell and activate the receptors. We have the postsynaptic receptor. So I always say one classical is the myasthenia gravis. You have the receptor deficiency. That's why you have the muscle weakness. Now this is very important. Endocrine. You have endocrine cells. Am I right? You release. So you can imagine you release the hormone. You through the bloodstream, and then you go to the target cell. So what's the advance of this? You can think about this. You can go to the very long. Am I right? So we have the pituitary. All the hormones through there, you through the blood volume, and then you can do all the target as the renal, you go to your, your gastro, and you go to the reproductive system. So that's the endocrine, the cell receptor. So an endocrine cell secretes signal molecule called the hormone that enters bloodstream and distributed widely throughout the organism. So endocrine signals can affect any cell that express receptor to the released hormone. So in summary, we just have 4 types. So one is contact dependent. The second is very local. Paracrine is also very local. And then synapse a little bit longer because remember you can neuromuscular junction and you see the nerve is very far. And the endocrine you can reach the whole, the body system surround. So this is the 4 types of the. So what's the chemical signaling mechanism? Synapse is targeted chemical release. Paracrine is local release of chemical just nearby the neurons. Endocrine release into bloodstream as a distant cells. All right. So the synapse, you have the vesicles, this acetylcholine, and then you open the calcium action potential through the sodium, and then you have calcium moving, you release, you activate this recept, and then transmit the action potential again. And the paracrine is just very locally, your target molecule is there. But endocrine, you go very far because through the bloodstream. Any questions? Just let me know. We talk about the chemical signaling mechanism. So for example, you have the signaling cells, right? You transmit the signal, you contact the receptor, and then they have a response. So the molecular signals such as glutamate, receptor molecules such as AMPA or NMDA, we have all these different kind of receptor, targeted molecules such as MBA channels. So the targeted receptor molecule could be the same, could be just the receptor channel. Am I right? There can many steps. There are many steps between the receptor and the targeted molecules. So the beauty of this is, I remember, we talk about this electrical synapse and the chemical synapse, all these different. Remember, electrical synapse, there's no ability for amplification. So this is the important concept. If you have the amplification, you have the receptor, you through the G protein. At this stage, no amplification, but through the ADC, amplified. Here, no amplification, but through the protein kinase, you also amplified. And through the phosphorylated transfer target protein, you see the amplified. You see the one signal, once through the amplification, exactly significantly, for the the neurochemical reaction is just one single molecular amplifies through the G protein, and through the CAMP, through the protein kinase, and through this amplified the signal. So if you do the electrical synapse in low, the interpretive animal, they don't have this kind of sophistication to do the amplification there. So amplification means each molecular receptor current may produce many G protein, and each G protein bound to the enzyme may produce many secondary message molecules, and they can be multiple many time scale. And this, this is there are 2 types of time scales. With ionotropic response, they are very brief, and the metabotropic response are slow but and long lasting. All right, so there are 3 classes of the cell signaling molecules. One is cell impermanent molecules. What does this mean? You have the transmembrane receptor. You do not cross it. Am I right? You have the signal molecule. You cannot permit it to go through there. That makes sense? So these are cell impermanent molecules. We will do some example there later. So you have the signal. This is cell permanent molecule. This is the cell molecules. You touch this membrane, you release, that can get in. And the other thing, they can go to the nucleus, and they have the intracellular receptor. So this is the cell permanent molecules. And they have cell associated molecules. So they come out. So this is just membrane to membrane. Receptor is there just at the surface of membrane, and through the receptor, then they change the signal, the signal, the new signal mechanism. So there are 3 classes of the cell signal molecule. One is the cell impermanent, the second is the cell permanent, and the cell associated molecules. So, for example, cell impermanent is a vesicle release bind to receptor. So they're including neurotransmitter and the neurotraffic factors. That's classical, like the acetylcholine, glutamate. Am I right? So cell permanent across the plasma membrane. Mainly, remember the membrane is lipid and on the water. Should be water insoluble. Otherwise, you cannot get in. Should be lipid soluble. Am I right? And there's special transporter protein, and that includes steroids and styrofoam. Okay? Yeah. So cell associated is on the extracellular surface of the plasma membrane. Only act on cell in physical contact, including neural cell adhesion molecules. So this is just cell contact molecule. So now in summary, this including like a neurotransmitter, like acetylcholine, glutamate, this is cell inflammasome. This is like steroids, thyroid hormone, and this is neurosignal molecules. So they do that. Am I right? Okay, so that's the three classes of the cell signal molecules. So we talk about these. So there are 4 types of receptor. First one is channel linked, ligand gated ion channel. We spent a lot of time. You remember my first, second, and third lecture there. This is a channel linked receptor. This is a channel closed. This is a channel open. You have the ions get in. This is the receptor. Once you activate, morphology, they change it. They leave it open. Am I right? So this is a channel linked receptor. Second type is enzyme linked receptor. So this is intracellular domain is an enzyme. So intracellular, this is intracellular, am I right? Should be intracellular, right? Active by the molecular binding to the extracellular domain. So molecular enzyme domain, am I right? Once you bind it, and then you have the cascade reaction. So this is they have the receptor RTK, tyrosine kinase, and neurotrophin and growth factor. Second type we call the enzyme linked receptor. And the third type is G protein coupled receptor. So you have the receptor there, and you have the G protein in the here. Am I right? So what's the example? Beta adrenergic, metrotropic ACH, M-glutamate, dopamine, GABA B. You have the receptor there. You have this this is neurotransmitter, and once they bind it, you see G protein change, and then you have the signal start the signal transmission there. Am I right? So the final one is we call the intracellular receptor. So what does this mean? You have the signaling molecules there. You go there, you get in. What's the example? We just talked about what kind of thing? Steroids, or hormone, thyroid hormone. Am I right? So you see this is a receptor. And once you bind the receptor, morphology is changing, and then you have chemical signal. Am I right? So what does this do is activated by the cell permanent molecular inhibitory protein complex disassociated when signaling molecule binds. DNA binding to domain revealed, active molecule move into the nucleus, and then the new mRNA transcript. So that's the the fourth type. So in summary, we have we just have the 4 type is channel linked, like the receptor antibody, enzyme linked, the G protein, and the intracellular receptor. So that's the the cellular receptors. We have 4 types there. Okay. So G protein, what's the G protein do? GTP binding proteins is binding to the GTP or GDP, and the GDP bond form is inactive. GDP bond form is inactive. GTP bond form is active. And there are 2 types. One is heterotrimetric. There are 3 different subunits. And the second type is monometric. I will show you some picture. They have 3 subunits: alpha, beta, and gamma, and it binds to the G, all the in summary, G, N, T, SA. Guanosine nucleotide, the GTP, or the GTP, many different subtype, and then the beta and the gamma bind to the alpha subunit. They stabilize the G protein in the membrane. They block alpha from interacting with the effector, and it can be the effect itself. So here we are. So let's look at the process this. So this is the chemical signaling molecule, and this is the receptor, all right? Once you binding there, what happened? This is the GDP, and this is the G protein. So you see the alpha, this is separate, am I right? This association. And then the GTP go to the GDP.
And then GTP replace GDP and then alpha dissociate.
Yeah, dissociate with it, yeah. And then what happened? Here, this is a subunit.
There's a adenine cyclase.
Yes. And then, no, this is effector protein. And then they go back to the recycle again. Am I right? So this is the heterochromatic GTP binding protein. So what happened? Binding of the neurotransmitter to the metabotropic receptor, activation of the GTP binding the protein, either the alpha or beta GABA subunit, diffusion of the G subunit within the membrane, and then activation of effector protein, and then you can activate the directed ion channel, or you have it through the enzyme and producing the secondary message system, and specific to the G protein subtype. All right, so what happened here? This is the monometric GTP binding protein. So you have the signal there. This is the same, am I right? So you look at it differently. Anyway, can explain this better. So you relay the signal from the cell surface receptor to cycloskeleton. This is cycloskeleton, okay? So vesicle trafficking 5 subfamilies. RAS is involved in cell differentiation and division, termination of the signal. So basically, the GTPase enzyme activates the G protein, GAP, hydrolysis of the GTP to the GDP, and the GAP for both types of G protein. So you have this activity, this inactive GTP for energy, and then you be the RAS be active, and then you go to the GAP. This is phosphorylation. All right. So this is a very important effector pathway of the G protein coupled receptor. So you go from here. So this is a very nice summary. You have neurotransmitter. You go to the receptor. You go to the G protein. Am I right? G protein binds to the enzyme. Then the effector protein. What happens? That enzyme produces intracellular diffusible secondary message system. Once you have the second message diffused into the cytosol to target the protein, okay? Then the ion channel kinase phosphorus this effect cell, and you go to the target action. So that's the effector, the pathway of the G protein coupled receptors. So here you can do like this. You have the neurotransmitter, you have the receptor, you have the G protein. If you go to the enzyme, this enzyme including ADC or the phospholipid C, they can through the second message system. They also can activate calcium, a cyclic nucleotide, phospholipid metabolics. So they can have the later effector and have the target action. This is the second type, am I right? Mm-hmm. And so this is the other type, effector pathway of the metabotropic glutamate receptor. So neurotransmitter is a glutamate, am I right? So you have the receptor here. Once you bind this G protein morphology change, effector protein is the phospholipid C, and then second message will be the IP3 and the diacylglycerol, and then later effector is the protein kinase C, calcium release. The target action is increase the protein phosphorylation and active calcium binding proteins. So, any question for this? So the calcium can also, act as a second messenger with a wide range of actions, including, second messenger with a wide range of, including the kinase phosphorus enzyme. We just mentioned the ADC, phosphorus, the ion channel, and the calcium release channels. So calcium has multiple sources. So we already talked about a lot with voltage-dependent calcium channel, the synaptic channel, NMDA channel, calcium release channel in endoplasmic reticulum, and the IP3 receptor. And that's activated by the second messenger IP3, rhodonidine receptor, activated by calcium positive feedback layer. So which receptor, this receptor is important in what kind of neurological condition? Yeah. One neurological condition is related with this. Any guess? No.
Yeah, one of the.
One of them is associated. Yeah, right. Yeah. No. The one is neuroleptic malignant syndrome. Yeah, it's related with that. With this rhodonidine receptor, I see. Yeah, but you're right. My senior graphs is the one thing related to that. Yeah. So calcium, okay, you have the multiple things. The binding protein is a buffer, K modulating activity, K binding inactive. There are the pumps and the calcium ATP cell, plasma memory, and plasma reticulum, and the sodium calcium exchange, diffusion, and the calcium is also in mitochondria. Calcium buffers. Calmodulin is a major calcium binding protein. Binds 4 calcium ions per molecule. They have a high affinity for the target enzyme. So this is including CA-MK2, CA-KIM5, and phosphodiesterase, AC, and PP2B, kind of the buffer there. All right, so this is we're talking about the second message system. So let's look at it step by step. You have the ADC, am I right? So, and then the CAMP produced from ATP, am I right? Once you're binding there by ADC cycles. So no question from here, am I right? Which is activated by GAS-GMP. CGMP is here, am I right? They are produced from GTP. What is GTP? It's not there.
Right at the arrow. The star green.
Star green?
The green is the stars. Or the spikes.
All right, yeah. Okay. Star green. That's the GTP. I'm sorry. Yeah, and then target is the kinase, the PKG, and the channel is CNG channel. So that's what inactivation by the phosphorylates. So this is second message, CAMP second message. Let's do this. So what's the PIP2 this? You have the PIP2 binding there. So the DAG and the isotope and the triphosphate is produced from the phospholipid phosphodiesterase is just by this, activated by G alpha and what say this Q. Okay. And then IPS3 activates the IPS3 receptor through the calcium release. DAG active protein kinase C inactivated by enzyme degradation. So that's this castle, PKC calcium degradation and the channel open. All right. So neuronal second message is in summary, we have the CAMP, CGMP, IP3 DAG. So this is the source, and this is the intracellular targets, and this is removal mechanism. We just did the summary, like this. And to be honest with you, after this, I forget immediately. When I was a medical school, when I was a medical student, I able to remember all this, to cram for the test, for this. This right now at my stage, I just forget it in 5 minutes. So there's no way I remember all this. There's no way this is kind of information I can remember. What's the AMP, GDP? It's so confusing there. So, but that's whatever, But this will be on your test. Medical student, you need to remember this. All right.
And there's drug targeting on this pathway.
Yeah, all this drug, right? Yeah. So the second message system, they have the spatial temporal activity pattern where and when are second message activated, control activation, and then the downstream positive. But I can also for all those, like the voltage gated L channel, action potential, how they generate it. This kind of thing I will remember for the rest of my life, because this is again and again reinforced. This is just a basic of neurophysiology. But this is a little bit, it's too much chemistry, I just don't remember. How action potential and the transmitter, this is the kind of stuff, you never forget. So the kinase and the phosphatase is in a phosphorylation state of the protein modulate activity depends on the balance between the kinase and the phosphorus. Phosphatase and the phosphorylation types including enzyme, ion channel, structure, and the regulatory protein, and mediate plasticity involving in the learning. So this is the regulation of the cellular protein by the phosphorylation in the second message system, ATP to the ADP, and the protein and the phosphorylation, and then you slow the protein phosphorylation, and the second message can do both sides. The kinase, and just put the phosphate group onto the amino acids. There are two major classes, serine and serine, active by the G protein coupled receptor, the second message. Tyrosine enzyme linked to the receptor, activated by extracellular signal. And each kinase regulates a particular sequence of amino acid on the many proteins. And regarding the kinase, each molecule has 3 essential domains. And the catalytic domain is activity, ATP and substrate binding domain. And and they have auto inhibitory domain and block catalytic domain. They also have the activator and the second message binding domain, and the binding of the activator causes conformational changes, and auto inhibitory domain move away from the catalytic domain. And we also, in the summary, the protein kinase A, and the structure is a cherometric protein, a dimer of regulatory subunit, and have the R subunit, and they have 2 CAMP binding site per subunit, and 2 catalytic subunit. They have the C subunit, and the ATP and the substrate binding site. The action of this is the binding of the CAMP reduce the affinity of the R4C subunit. And the C subunit dissociation. You remember this? R inhibitor acting when by the R subunit. All right, so we talk about the calcium modulating dependent protein kinase. I think I just skip that. I think that nobody will remember this. Yeah. So this is the protein kinase C structure and activation. Catalytic region near the C terminal, regulatory near the N terminal, and the auto-inhibitory domain in the middle, and they have several binding regions, including GAG and the PS and the calcium. So tyrosine kinase, they have membrane binding protein. They have extracellular binding domain, and including growth factor, neurotrophic factor, and intracellular cataract domain. And the non-receptor tyrosine kinase have the cytosolic and the membrane associated indirectly by extracellular signal and involved in cell growth and differentiation. All right. So this is something back to the long-term depression and a long-term potentiation. Anyone know the long-term potentiation? Memory related. Joe, what do you know?
I know the mechanism.
Yeah, you remember what kind of experiment they did? Long-term potentiation.
I guess I don't know.
Yeah, by the Eric. Yeah, make it stronger. That they proved that, and they got the Nobel Prize used by Eric Kandel at Columbia University. They study what animal they do.
Monkey?
I know it's
No.
the study you're talking about, but like.
Oh, dog?
No, not dog.
Not a condition.
Yeah, not the condition.
So the heavy connectivity, so like
Yeah.
When the
Yeah. It's not just software. Some small.
It has to be higher level.
No, low level.
It's
Low level, very low level. Long-term potentiation.
Intelligence?
No.
Yeah, something like that. Like they develop like that. So around this, but I don't know if this is one.
It's done by Eric Kandel.
Yes.
At least they say hippocampal slices.
And maybe hippocampus size. Yes. All right.
A rabbit?
No, I don't think so. This is a low. Yeah. So this is the long-term to the cerebellar synapse. So this is the cerebellum, and the major one is I know this guy is Masao Ito from Japan, and I met him personally. So that's the one. Yeah. So that's why I know this very. That's why I know him. Yeah. So Masao Ito, and So cerebellum help coordinate the motor movement. Prepare fiber synapse onto the cerebellar Purkinje cell. the frog release the glutamate binding to the AMPA receptor causes small local depolarization. And the climbing fibers synapse onto the cerebral ganglion cells generate and release glutamate binding to the AMPA receptor causes massive depolarization and the calcium influence. So LTD means long-term depression, of the EPSP. So this is the papiral fiber release glutamate, stimulate N-glutamate, alpha and Q active the PLC. PLC produces the IP3 and the DAG. DAG along cannot. So that's just a full pathway there. And this is the cerebral synapse. So what does this mean? So I think I just found this from the internet. What does this mean? In the hippocampus, we're talking about long-term potentiation. So what happens is you have the calcium kinase activation. You have the long-term potentiation. And that's because it increases the synaptic AMPA receptors. If you decreasing the synaptic the AMPA receptor, the calcium getting in the long-term depression. So I just want you understand this, okay? Now look at the hippocampus, okay? Remember, CL1 neuron is very important for memory, right? CL is very important to the memory there. If you have the calcium getting in, if you active the kinase, if you have the kinase activation, is a long-term potentiation. If this is the calcium you get in, if you slow the phosphorus activation, is long-term depression. The same calcium, you active different signal in the neuron, produce a different.
But this depends on the concentration of calcium.
Yes.
Only high concentration of calcium will cause long-term potentiation, but the other way.
Yeah, but in the cerebellum, just opposite.
Oh.
In the cerebellum, the papiral fiber and the Purkinje cells help produce decreased synaptic activity. So this is the kind of thing could be the opposite. So this is the calcium, the concentration, right?
Yeah.
So that's just a different, So that's the depression, long-term potentiation, long-term depression is remember memory. That's how the neuron remember things. After you stimulate it, they remember it. Next time you have a small stimulation, you can produce. That's you're exactly right. For example, the first time you need to 100, let me say the calcium to stimulate it. After you learn, next time 5 or 10 will be enough to activate the same response. You remember it. So that's all about. That's all for this signal. I think that's the last one. All right. Thank you very much.
Thank you.
Yeah, I will email you. All this.
It's very detailed.
I have dementia myself. This is just.
Because if you have dementia, why do we have?
I was very fortunate in all those pathways. It's impossible. Kindness, force, what are they?
So many.
Cells now.
Yeah.
Yeah.